Hello and welcome. You are listening to a podcast by the Milner Centre for Evolution at the University of Bath. I'm Professor Turi King, your host, and today I'm talking to Nick Priest, who is lecturer in evolutionary biology, about his new research identifying the multiple drivers of cactus diversification, which is out in Nature Communications. Now, Nick, before we talk about this new research, I know you had a really interesting paper which came out last year that was looking at the diversity in orchids. And I know Darwin also studied orchids, and he proposed that orchids had evolved over a really long time with species adapting to attract different pollinators. But what you and your team found showed that that wasn't actually the case. So talk me through, what were you looking at and what did you find?
In the middle of the COVID pandemic, we had a huge shift in what the lab did because we couldn't do our research. So we came up with this project looking at diversification of this group of plants that Darwin had worked on. And what my PhD student, Jamie Thompson, found was that when you look at the phylogeny of a group of orchids, the terrestrial orchids, which it turns out was the group that Darwin was so fascinated with, there's a very conspicuous pattern in that most of the species appeared to arise fairly recently. And when you look across the history of the planet Earth, the pattern of the relationship between different species, it's very conspicuous in the orchids. Why did so many arise so quickly? And it turned out this was the big dilemma that Darwin was facing all those years ago. And it was sitting there right in front of us in the terrestrial orchids.
So basically what you've got kind of like a family tree, essentially, of terrestrial orchids. And you're looking at, like, when are these branches arising? And you were finding that a lot of them arise all at once. Why is that happening?
Yeah, so once you have a phylogeny, then it turns out with modern statistical methods, there are ways of testing what are the possible causes to explain that pattern. And of course, the big one we had to exclude was the one Darwin thought, that they must be around for eons. And so it must have been inconsistent with just a gradual evolution of different species. And by testing different hypotheses, for example, changes in carbon dioxide in the Earth's history, or even the well-known changes in sea level that happened, we could then exclude a number of hypotheses. And through other kinds of tests, we could work out that it really had to be temperature change in the Earth's history that drove it, not other types of climate change or time or even pollinators driving the patterns across the whole group.
So how do you work out when this kind of sudden change happened? How do you work that out?
Yeah, I got to admit, this is one of the dark arts of evolutionary genomics. For a long time, we've known about techniques in molecular clocks that allow us to time when various events happen. What makes it particularly difficult in this group of plants, as it's true in all succulent plants or plants that don't leave a residue in the fossil record, is that we don't have rocks that we can use that we know happened at a particular time to sort of cement when it happened. In this case, we relied heavily on changes in plastid genomes to give us inferences. And this was considered a gold standard for explaining changes in orchids and other related plant groups. So by working very, very carefully with established timing points, we then had confidence of when these species arose, and that allowed us to then test which potential ecological hypotheses could be driving it.
So essentially what you can do is you can look at the DNA of various species. We know that there's genetic differences between them, and we know that those differences, those mutations that cause those genetic differences, they happen on kind of a regular sort of basis. So you can use them like a clock. So this is that molecular clock that you're talking about. So by looking at those differences, you can start to time as to when all that coalesces back to. And you found that this radiation happened about 10 million years ago, is that right?
The origins of the orchids date back much longer than that. But when we look at when did the big burst happen, the new species happen, it all happened really in the last 10 million years, which may seem like a long time to your listeners here, but actually in the eye of evolution, it's a blink. So,
and it's when climate change was changing, but not how we think it now, where we know everything is getting warmer. This was when stuff was getting cooler, wasn't it?
Yeah, this is the major ice age that people talk about, where major glaciers formed, and we can still see residues of those glaciers. In fact, A lot of the most depressing news stories we see are those glaciers falling apart that came about during that era, where the global temperature precipitously dropped for a whole number of reasons. And for plants in particular habitats who can't move, they're then trapped in environments that aren't as suitable for life. And we think it was that, probably with other patterns that we haven't yet explored, haven't yet explained, that then drove it. But it was certainly triggered by this cooling event.
And do we know why cooling temperatures did this? Or is that your next question? We can see that that's what's happening, but we don't know quite why it's happening. Or do you know why it's happening?
No, we have absolutely no clue. I want to give a shout out to my cousin, Gretel Kiefer, who turns out is one of the world experts in a single type of orchid. She has a 30-year data set of the Western Fringe Prairie orchid, and she's shown that this orchid, its seed set changes from year to year, and there might be related to changes in climate that she's observing. Now, if she can observe that in one species, that you see major variation happening in reproductive output, it's not too surprising that this could be simultaneously happening in many, many species at the same time.
This is what you were finding in orchids, and then you kind of turned your attention to cacti, which I didn't realize is one of the most endangered of any taxonomic lineages. So tell us about cacti. So what's the starting point for your research when you're starting to look at cacti? You're wanting to know about what's driving diversity. What was the thinking at the time?
Yeah, I got to say, most of this is because we thought cacti are cool. and as a group, it fit the same thing as the orchids in that it has a huge number of species and they seem odd. But once we had information that orchids could be driven by climate change, it made us think, well, what about other groups? And if we dug down deeper, could we find specific aspects of climate that are driving the diversification of other groups? And the cacti just seemed like a great group. One thing I want to add to that is there have been lots of hypotheses about what could be driving diversification in cacti. From my quick look through, I found at least 25 different hypotheses out there that actually seemed reasonably well supported. But of course, it doesn't make sense that so many things could be driving diversification. And so we had to think carefully about the approaches we used to then test patterns in cacti, and not just be another one throwing up another hypothesis, but trying to do it more concretely.
So what did you do?
Yeah, so we turned to the power of artificial intelligence. And there's some features about AI that make it really good for testing lots of hypotheses simultaneously. It just has an efficient configuration. So a major problem in evolutionary biology is we just don't have the computer power to understand shifts across entire phylogenies with thousands and thousands of species across thousands and thousands of traits in thousands of locations. It's just a computational problem. But machine learning is set up to make that very efficient. And then the other neat feature about machine learning is it allows you to specify the level of complexity that you test with the data set. So we could address a whole set of different hypotheses simultaneously and test for what are the effects of each of them individually. But then we could also just change the dial to allow us to then test for how do these factors interact, and could it be multiple forces driving? And if you consider multiple forces working together driving diversification, then it might actually be a different list that we would identify as being important for diversification.
So what are the various things you're kind of trying to feed into this to find out which has the most influence?
Yeah, well, I got to give a lot of props to my PhD student, Jamie Thompson, about this, because he quickly realized that there were a set of different data sets that we could apply. And so in addition to having the phylogeny, we then worked out what are the climatic variables all across the Earth, and then bringing in other information about soil quality, geographic factors, on top of things like chromosome number counts. We can then assess a whole set of questions independently. Once we had built this big data set, then we could explore how those factors contribute to the evolutionary patterns we can see in the phylogeny.
So that's really cool. So you can feed in all of this information basically into a computer program that then goes, right, well, it's working out how these things are interacting and what seems to be the most important factors in driving speciation and diversity. And what did you find?
Yeah, so after all that, testing 39 independent hypotheses across the entire phylogeny, the three that came up were not what the experts would have predicted as being central drivers. We would have thought that it was pollination driving everything. And in fact, that was one of the major features that Darwin focused on. And it made complete sense and still makes complete sense. And it still may rue the day once we get better data, but it didn't pop up as being a central driver in our analysis. So it turned out the diurnal temperature range, so just what the plant experiences in its location and a day-to-day flexibility. When we think about cacti, they look the kind of a prototype example of a succulent plant, which has a fleshy surface that we know is really good at capturing water and maintaining water. And so we would have thought that it was due to the dryness in the air, the aridity, that must have caused that, because it seems like, oh, well, they're so good at capturing water. And so we thought, well, they must be diversifying where it's driest. But actually, they're in wet climates as well. And there's a huge variety of forms. And so we quickly turned to cactus experts for our help. And we were so fortunate to have Tanya Hernandez join in on the team. And she was a huge asset to giving us an understanding of the ecology and also why we might not expect aridity to be such an important driver.
So what are the other ones besides this temperature thing?
So the one that surprised us the most was the second highest rated variable, which was the sand content of the soil. And then the size of the plant came out. Now, the one thing about these machine learning analyses is when it pops up as something like, oh, here's the variable that's important, it doesn't actually tell you what the pattern is. And when we did some intense mapping onto it, it turns out it's very small cacti and very large cacti that diversify rapidly. And they may be diversifying for different reasons, but it was those two extremes that worked out, for example. And then just to add in that the isothermality, the daily variation in temperature, and then compare that to annual variation in temperature, it measures the ratio of the two. How important is an annual variation relative to a daily variation? And I think one thing that gives us some confidence in this is that it turned out it was an intermediate isothermality, which means that the daily fluctuations are about half of the variation in the annual fluctuation. So that seems about right in terms of it's not saying it's super extreme, and it happens enough across the cactus phylogeny that we're pretty confident that that's important. And the only other last variable was geographic size. It's a funny variable because it basically looks at how the coverage of species across vast terrain. So in some ways, a large geographic range kind of makes sense that maybe there's more opportunities to find special niches where then it can diversify. So it turned out that the geographic range also explained patterns of diversification.
So why do we care about what's driving evolution? So evolution is essentially where you get diversity. So diversity starts at the genetic level and then it's selected for. So there might be things that are making it more advantageous or disadvantageous in particular regions or areas or whatever. Why are we bothered about that?
Well, I think anyone that looks at the changes that are happening in our climate now does make you wonder what's going to happen. And what basic evolutionary biology tells us is if there's genetic variation, and there's variation in responses to selection, then you're going to get an evolutionary response. We're going to see a conspicuous change in the nature of the species we're looking at. That's something that microevolutionary research has told us. What we're trying to do with this kind of research is essentially map on the microevolutionary insights into the macroevolutionary patterns. So if we can identify the ecological drivers for macroevolutionary patterns, it can give us insights into those smaller changes happening within populations of organisms that are driving populations in particular directions that could then change organisms across time. So I think we care because it's how we got here, and we care because it's where we're going. And I think this type of approach that mixes insights from macro and the micro is sort of at the frontier that hasn't been explored much, and frankly couldn't be explored until we had broad data sets available to us and then the computing power to be able to test for it.
And you found that temperature is one of the main drivers, and that's obviously really important, as you've already alluded to some extent, with climate change.
Yeah, so you might say, well, what we learned with the orchids isn't relevant here because that's a global cooling, not a global warming, right? But it could well be that it's really about the temperature extremes. Or it could well be that extreme cold period then created an environment that wasn't hospitable for the pollinators, which then caused problems for the plants. I should say that although we've excluded pollinators as being the drivers, that's just what the evidence we have tells us. With better data, we might have a more informed picture. And I should point this out that in the case of the orchids, we were able to show that diversification is 700 times more likely to be driven by global cooling than just the time that Darwin had envisioned. But we still haven't explained all of biodiversity. And in the case of the cacti, it's more than 70% of the variation is unexplained, which means we either need better data sets or better methods in order to work out what's actually been the major factor driving it. We're chuffed to bits that we were able to identify important drivers, but there's still a long way to go.
So, Nick, what's next for you?
We've established now that there is this effect of global cooling on orchids, and there's climatic effects on cacti. We're also investigating general effects across all succulent plants, trying to work out the timing of the evolution of succulents. But it still gets back to this general question of, now that we've learned so much about climate, can we apply it to tell us something about whether we are at a tipping point in evolution? And Jamie and I have had a thought of putting together a collaboration of scientists to get at whether these tipping points are happening across lots of different groups of plants. One of the ones I'm most interested in is the ferns. So working with ancient groups of plants and studying their patterns of diversification might give us more information. So what might be possible if climate has had such a big effect in evolution, if it does contribute to these tipping points, then we should see a residue of that in the fern phylogeny as well. And what we'd like to do is build models that allow us to identify not just global cooling events, but global warming events as well. And then identify the temperatures at which those bursts in speciation and extinction happen. So if we had a catalog of hundreds of plant species and when their big diversification events happen and how that relates to climate change, then it might give us a lot more confidence about whether the global warming we're now experiencing is a serious problem, or actually we still have another 5 degrees to go until the species start crumbling. I think this type of research is something that there's a lot more active engagement in, but I do think our study contributed to showing the clearest pattern of temperature-driven diversification to date. And I'm hopeful that we can build a team together to then assess this across all plant groups.
Nick, thank you so much for talking with me. This was a podcast by the Milner Center for Evolution at the University of Bath. I'm Turi King, and thank you for listening. If you have any thoughts or comments on this or any other episodes, please contact us via our X channel @Milner Center. For more information about the Milner Center for Evolution, you can visit our website.